Ishir, thank you very much for the kind introduction, and thank you, Francesca and Felix, for inviting me for this fantastic seminar series. I'll present a work very much in progress, and the key message of my talk is already written in the title of the first slide. So Golgi quality control preserves cellular architecture, and I'll try to explain you how we discovered two different branches of Golgi quality control and how we're trying to characterize how they maintain Golgi architecture and cellular architecture together. My lab started out by being interested in pathways that specifically degrade membrane proteins. And as you are well aware that at the ER, where most membrane proteins are born by being co-translationally inserted, the first quality control mechanism is the ER-associated degradation system. And this ER-associated degradation system recognizes misfolded proteins, retrotranslocates them out of the ER membrane, ubiquitinates them, and then feeds them into proteasomes for degradation. Once membrane proteins are properly folded, they can be exported from the ER into the Golgi, and once they're exported from the ER, it is much less clear how membrane proteins are ubiquitinated and targeted for degradation. But for a long time, it was thought that the major or the only degradation pathway that degrades ubiquitinated membrane proteins are the endosomal sorting complexes required for transport, which recognize ubiquitinated membrane proteins on endosomes and then catalyze a negative membrane curvature to generate intraluminal vesicles and thereby drive the biogenesis of multivesicular bodies, which once they fuse with lysosomes, deliver these intraluminal vesicles that carry the membrane proteins, and therefore these membrane proteins can be efficiently degraded in lysosomes. And the escort machinery originally was discovered by Scott Emmer, and Wes Sundquist then identified or found out that escorts are also required for HIV budding from the plasma membrane. And then year after year, new functions for the escort machinery were discovered. And we contributed a few functions, and escorts seem to be everywhere. They seem to be doing everything. And, you know, they have functions at the nuclear envelope, they have functions at the plasma membrane, and they're important for MVV biogenesis. So when I was already a postdoc in Scott's lab, I started wondering, how can it be that this important machinery is not essential for life? So in budding yeast, it's the model organism that we are using for our studies. You can knock out the escort machinery, and yeast is actually pretty fine. They don't care very much. You know, they're a little bit heat sensitive, but otherwise they grow pretty normally. And this is astounding, because if you look at the morphology of escort mutants, and you follow a GFP-tagged membrane proteins, either in cells where the escort machinery is active, you can see that this GFP-tagged membrane protein is nicely sorted into the vacuole, which is the yeast ortholog of lysosomes, through these multivesicular body pathway, right? These are electron tomograms of multivesicular bodies, and you see that they have this limiting membrane and the intraluminal vesicle, and every membrane protein that arrives in the lumen of the vacuole has to go through these intraluminal vesicles. But if you knock out the escort machinery, any component of the escort machinery, you observe two striking phenotypes. First of all, membrane proteins are no longer sorted into lysosomes. They get stuck on these multilamellar class E compartments, which form instead of the multivesicular bodies. And they're always stuck there, and we never see them disappear. So every membrane protein that's supposed to be degraded ends up in that compartment. So we wondered, how can it be that cells can survive with, you know, such a rock essentially that sits in their cell and accumulates all these ubiquitinated membrane proteins. So being in Scott's lab and being a geneticist, I thought what we'll do when I start my lab is we will do genetic screens. And we'll just ask, are there components in the yeast genome that are required for the survival of escort mutants? And if so, what are these components? So this is what Olli Schmidt, the postdoc in my lab, who is now an independent group leader, started to do. He simply crossed escort mutants against the entire yeast knockout collection and tried to identify which genes were essential for the survival of escort mutants. And the screen returned 119 synthetically sick or synthetically lethal mutants. And against my expectation, The screen did not return components of the ER-associated degradation machinery. It did not return components of the unfolded protein response, and it did also not identify autophagy. So all of these pathways are not essential for the survival of escort mutants. Yet it identified 3 out of 4 components of a ubiquitin ligase complex that is referred to as the DSC complex. And DSC stands for defective in SREBP cleavage complex. And to cut the long story short, because this is all published data, what Oli could show is that the DSC complex, which sits at the Golgi in budding yeast, can recognize a repressor of single lipid synthesis that is exported from the ER. And once this ER protein reaches the Golgi, the DSC complex ubiquitinates this repressor of single lipid biosynthesis. And this ubiquitination does not lead to escort-dependent degradation of that membrane protein, but to the extraction of this membrane protein from the Golgi and to the proteasomal degradation of this membrane protein. So with this single cargo, we proposed that we had identified a novel protein degradation pathway that we termed endosome and Golgi-associated degradation, or short EGAD, that Ishe referred to already in his introduction. The DSC complex consists of 4 core subunits. It consists of a ubiquitin ligase that is called TAL1, that has a ring domain in its cytosolic domain and a large luminal domain. It consists of a rhomboid pseudoprotease that is called DSC2, of a protein that is referred to as DSC3 that contains a ubiquitin-like domain, also in its cytosolic domain, and of UBX3, which harbors a UBX domain. So this complex composition is very reminiscent of ERAD E3 ubiquitin ligase complexes. They typically have a very similar composition. They also have rhomboid pseudoproteases and CDC48 or VCP recruiting enzymes such as the UBX domain. So what Yannick and Diduna, Yannick is now a postdoc in my lab and Diduna is currently writing up her thesis, wanted to figure out were 2 questions. Question number 1, which we just published, is how does the DSC complex identify its substrates? How does it find proteins at the Golgi, ubiquitinates them specifically, and targets them for degradation? And what Yannick could show by identifying additional substrates of the DSC complex, we now know of 3, or we now have characterized 3 substrates, ORM2, YIF1, and HMX1. All of these substrates are ER proteins that for one reason or another are at some point exiting the ER and arriving at the Golgi. At the Golgi, the DSC complex uses its rhomboid pseudoprotease subunit DSC2 to thin the membrane and to detect preferentially transmembrane proteins that have short transmembrane domains. And once that interaction occurs, these proteins are polyubiquitinated, and they can either be K48 polyubiquitinated, and then 2 substrates, ORM2 and YIF1, are exclusively extracted by CDC48 from Golgi membranes and fed into proteasomes for degradation. In the case of HMX1, it's a bit more complicated. So HMX1 is a transmembrane protein that must be ubiquitinated by the DSC complex, but once it is ubiquitinated, it can either be extracted by CDC48 or it can be passed on to the escort machinery for lysosomal degradation. How this discrimination between the substrates happens, we do not understand very well. But what we do understand now is that the DSC complex uses a process that we like to refer as hydrophobic matching or hydrophobic mismatching to identify its substrates. So it likes to interact with proteins that have a short transmembrane domain and that are preferentially ER proteins that have been mislocalized or orphaned from the ER to the Golgi. And there it identifies these proteins, targets them for degradation, and therefore prevents their spreading across other cellular membranes, in particular endosomes and lysosomal membranes. So once we understood this process a little bit better, Iduna started to ask, are there other components to this Golgi quality control system? And are these components, together with the DSC complex, perhaps essential for the survival of yeast cells? So we conducted another genetic screen in which we, together with the Corman lab in Oxford, used an approach called SATI, which is saturated transposon analysis in yeast. So we used DSC mutants and randomly inserted transposons across the entire genome, saturating the genome by having transposons insertions every 42 base pairs. And then we just mass sequenced these libraries and asked which genes were enriched or depleted in transposons. Those that were depleted in transposons insertions were genes that were essential for growth and survival of DSC deficient cells. And the top hit from that screen, which was essential for the growth of DSC mutants, was a protein called RER1 or RER1. And RER1 is essential for growth of these cells, and it is also upregulated in DSC mutants on protein level, already suggesting that they could work together. Then we confirmed this interaction between the DSC complex and RER1 by simple liquid growth assays, and you can see that wild-type cells or DSC mutants grow well, but if you knock out RER1 in addition, they grow very, very poorly. And at the same time, they lose the integrity of their plasma membrane because now propydium iodide enters these cells. So the interaction, the genetic interaction between RER1 and the DSC complex is essential for cell growth, and it maintains plasma membrane integrity. And then we asked, so RER1, does it specifically interact with the E3 ligase at the Golgi, or does it also interact with other quality control systems in the cell? But it does not. So you can knock out RER1, and at the same time you can delete ERAD E3 ligase, and you do not see a genetic interaction. And you can do the same with autophagy. So this interaction, this genetic interaction between RER1 is specific for Golgi quality control processes and not for ER quality control processes or autophagy. So what is RER1? RER1 is an evolutionary highly conserved ER retrieval receptor that cycles between the ER and the Golgi and picks up orphan proteins from the Golgi and returns them back to the ER. And one major substrate of RER1 is Sec12. Sec12, as probably most of you know, is essential for the activation of SAR1 because it's the gap for SAR1 and therefore essential for the formation of COP2 vesicles at the ER. And it leaks continuously out of the ER and needs to be returned by RER1 back to the ER to retain it there. Hence the name of RER1, retention for ER proteins. So then the obvious question from that genetic interaction was, do RER1 and the DSC complex function together to prevent the accumulation of orphan proteins at the Golgi or in other post-Golgi compartments? So to test this idea directly, we simply conducted quantitative proteomics experiments and measured which proteins would accumulate when we inactivate RER1 and TAL1 at the same time. So we essentially switch off both these two components for Golgi quality control. And remarkably, we found that 51 membrane proteins were strongly upregulated in these double mutants. Overall, we identified 600 membrane proteins, which corresponds to approximately 50% of all yeast membrane proteins, which is pretty strong for identifying quantitatively on a proteomics level membrane proteins. And so approximately 8% of all membrane proteins were upregulated in these double mutants. And of those 51 membrane proteins, we identified 16 ER proteins, 4 Golgi proteins, and the remainder were membrane proteins at the plasma membrane, the vacuole, and endosomes. So across the board, membrane proteins appear to accumulate. And they accumulate specifically in the double mutant, but not so in TAL1 single mutants. And reassuringly, Sec12, the main cargo for RER1, was also upregulated in these hits, suggesting that you need the DSC complex to degrade Sec12 once it escapes from the ER. And that can be demonstrated nicely by live cell fluorescence microscopy. So in wild-type cells, Sec12 localizes to the nuclear and the peripheral ER in yeast. In RER1 mutants, Sec12 exits the ER and is efficiently delivered into lysosomes. But if you have a TAL1 RER1 double mutant, RER1 can still be detected at the Golgi and on these additional rather blurry compartments throughout the cell. So together with the proteomics data, these findings suggested that the DSC complex and RER1 together prevent the accumulation of orphaned membrane proteins on post-ER compartments. And we have similar results for the other cargoes that we identified. ORM2, HMX1, and YIF1 also accumulate on post-ER compartments in the double mutants. So then we went on to characterize what would happen to the Golgi under these conditions when Golgi quality control collapses. And we characterized early, medial, and different late Golgi markers. And for each of these markers, we found that the number of Golgi compartments increased once Golgi quality control collapsed. So early, medial, and late Golgi were more in numbers, suggesting that the Golgi expanded once these proteins started to accumulate on post-Golgi compartments. This is somewhat reminiscent of what happens when the ER is being stressed and unfolded proteins accumulate in the ER. Also there, the cells initiate a response where the ER is becoming larger. So from this data, we concluded that once Golgi quality control is active, cells can maintain a proper distribution of proteins. They either return orphan proteins back to the ER or they degrade them at the Golgi. But once these two branches of Golgi quality control collapse simultaneously, these orphan proteins begin to accumulate at the Golgi and spread throughout the cell. And at the same time, the Golgi expands and you generate more Golgi-like structures in the cells. To really test whether this was true, we then used electron microscopy, high pressure freezing and electron microscopy. And we saw a massive disorganization of the affected cells. So here you see yeast cells, high pressure frozen and analyzed by transmission electron microscopy. And what we observed were these very large, very strange looking structures in these mutants. And you can also see that other organelles were also massively disorganized in the mutant. And what was interesting and what we see over and over again, but we did not have time to analyze this properly, is that the daughter cells, this is a budding daughter cell that grows out of this mother cell, is actually pretty clean and does not look very disturbed, as if the majority of the damage would be retained in the mother cell. But this is something that we have not yet fully characterized. All right, and then we went on to characterize the cellular phenotype. But I think from the data that I've shown you so far, it's fair to conclude that Golgi quality control is essential to maintain subcellular architecture. And then we went on to characterize Golgi, and we thought we could look at PI4P, phosphatidylenosytol 4-phosphate. And we were in for a very big surprise. So when you look at PI4P using different PI4P probes, so either the P4C probe, the pH domain of FAP1, or another PI4P binding probe, we observed always the same thing. In the mutants where Golgi quality control is collapsed, we saw these huge structures that are ring-like or sacculus-like that accumulated in these cells. And they're all enriched for these PI4P probes. So the FAP domain binds to these probes only in the presence of active ARF1. The K5 loop only binds to PI4P in the presence of phosphatidylserine and ergosterol. So we figured something important must be going on, and we wanted to understand what these huge structures are. And we teamed up with Florian Wilfling and his lab and performed cryogenic correlative focused ion beam milling and cryo-electron tomography. Oh, and first of all, I want to show you that also ARF1 is on these compartments, as already predicted by the presence of the PI4P sensor. If we look at GFP-tagged ARF1 in wild-type cells, it localizes at the Golgi with some of these PI4P dots that are picked up by the PI4P sensor. When Golgi quality control is collapsed, ARF1 is on these very large structures that are PI4P positive. And these are not lysosomes. I can tell you that right away. So, and on these compartments, it's important to note, we also detect PIK1, which is the PI4 kinase, deacylglycerol, neutral lipids, and of course, PI4P. So overall, they have a lot of the components that would make up a trans Golgi. But when we looked at them by electron microscopy, we saw something that we did not expect at all. So when we do correlative electron microscopy of these PI4P containing rings, we observe this massive accumulation of circular membranes. And we did not observe them once, we observed them in almost every cell that has these PF4P positive structures. And if you look very closely at these structures, you can see double membranes that are in very, very close opposition to each other, sometimes touching each other almost, but not apparently not fusing with each other. So what these structures exactly are, we do not understand, but we have decided to call them for now Golgi-derived compartments, or GDCs. I want to show you a little bit more on these GDCs. So what we immediately thought when we saw these multilamellar compartments, we thought, this is going to be easy. It's like in escort mutants. You form these multilamellar compartments, like in escorts, and like in escorts, all the proteins that accumulate will accumulate also in these multilamellar Golgi-derived compartments. But we were completely wrong. So all the cargoes that we have looked at are excluded from these compartments, and they're not transported in these compartments. And we have not just looked at HMX1 or SEC12, which accumulate here, but never colocalize with these huge PF4P positive structures. We have looked at 20 other markers so far. Also, a lot of Golgi markers are not on these, what we call Golgi-derived compartments. So we really do not understand very well what they are and what they do. But what we do think is that they are very important for the survival of these cells. Because if you knock out ARF1, these compartments completely disappear. So here we label the compartments with body P. You can label them with body P because they are rich in neutral lipids. If you delete ARF1, these compartments are completely gone. But the cells now grow very, very poorly. They're almost not alive anymore. Now we can just barely keep them growing. So somehow ARF1 is essential for the biogenesis of these Golgi-derived compartments. And what we also noted is that these Golgi-derived compartments are not dead-end compartments, but they depend on the growth of cells and probably on the protein load that you send through the secretory pathway. So in growing cells, you form these ARF1 positive GDCs, but as cells enter quiescence and stop to grow, ARF1 gets inactivated, relocalizes from the membrane, and now these GDCs are also no longer formed. So these organelles, these multilamellar organelles that are rich in neutral lipids, are not dead-end compartments, and somehow they respond to the protein load of the secretory pathway. So with that, I'm almost at the end of my presentation. What we believe is going on, but we don't have a final answer, so it's very much work in progress, is that we have identified a Golgi quality control network or system that prevents the accumulation of orphaned proteins during cell growth. We speculate that this accumulation leads to lipid packing defects at the trans-Golgi and to DAG accumulation, which in turn triggers ARF1 activation, the recruitment of the PF4 kinase PIK1 and PF4P synthesis, and in some sort of vicious loop, this leads to the generation of these Golgi-derived compartments. We also believe that these Golgi-derived compartments are important for the survival of cells. We just do not fully understand yet how they relieve the stress of cells when Golgi quality control has collapsed. So with that, I'd like to summarize. I hope I could convince you that we have identified an additional component that works together with the DSC complex at the Golgi, which is a retrieval receptor. So the DSC complex will ubiquitinate and degrade orphan proteins at the Golgi, and RER1 will retrieve orphan proteins. If one or the other is disrupted, cells don't care so much because they have this backup system in place. But if both of them are gone, then cells get really in trouble because now more proteins will become orphans. These orphans will accumulate and there is no way that you can take care of them. They start to accumulate and essentially start to poison the endomembrane system. And as a response, we believe as a stress response, these GDCs, these multilamellar compartments are formed and somehow help the cell to survive. So with that, I'm at the end. And if we haven't talked enough about Golgi in the first talk, maybe now there was a good dose of Golgi for all of you. All right. Thank you very much for your attention. Of course, I would like to thank the people who did most of the work. So Olli initiated the project, and Yannick and Iduna are carrying the project forward. And with that, I would like to also thank the collaborators, among them Chris Stefan, who has provided a lot of the PF4P probes, Benoit Corman, Florian Fröhlich, and the Wilfling Lab in Frankfurt for their support and for their help. And I thank you for your attention, and I'm happy to take questions and maybe some suggestions of what's going on. Thank you.